This is question 9, the last and final question of paper 2 AS Biology for 2017. Very little demand in the first part of this question as there's very little uplift from GCSE. With that in mind, we need to concentrate on the parts of A-level that have lifted it from GCSE and not give a GCSE answer. So it says, describe the cohesion tension theory of water transport in the xylem. Well, the first thing we're going to say is that water is lost through evaporation of water through the stomata of the leaf. This then lowers the water potential of the mesophyll cell. And that takes us into A-level territory because we didn't learn about water potential at GCSE. And also it's the idea that the water is evaporating from the surface of the mesophyll cell because there's all those air spaces between them because they have an irregular shape. Then it's the idea that because we've lost water from that cell and the water potential has lowered, we want to replace the water, and we're going to do that with water from the xylem. So we're going to say that water is pulled up the xylem, and that is the third mark in the question. And that pulling of water is the tension part of the theory. And water is pulled because hydrogen bonds exist between the hydrogen and the oxygen atoms of different molecules. That represents the cohesion part of the theory. And I do like the statement or the sentence, as water coheres together by hydrogen bonds. It's a lot more sophisticated than saying water molecules stick together. And this, of course, forms a continuous water column. Is the whole point of the cohesion tension theory of water transport. If there's a break in that column, then water will not be pulled up the xylem vessel anymore. And if we missed any marks there, by mentioning not just cohesion but adhesion, which is the sticking or the adhering of water molecules to the walls of the xylem vessel, would give us a possible 6th mark. Of course, that adhesion helps to support the cohesion. And then as we move on to the last question, it says, describe how mRNA is produced in a plant cell. Well, at this point, we shouldn't think that it's produced any differently than in an animal cell. I'm going to start off by saying that the DNA strands would separate by breaking the hydrogen bonds between the complementary DNA bases. Now, that's a real A-level answer. What we don't want to say is that the DNA unzips. In this mark scheme, it didn't discriminate. It didn't say you couldn't say that, but in other mark schemes, it has. So best to stay away from the whole unzipping idea and talking about the bonds, the hydrogen bonds between the bases breaking. There was then a mark for saying that only one of the strands, one of the DNA strands, is actually used. And then the third mark was to say that complementary base pairing takes place. So A joins with U, T joins with A, C joins with G, G joins with C. Now 28% of students gained no marks, and of those students who did score well on this question, most of them omitted this bit of detail here. Why I've circled it. The next mark was for saying that RNA nucleotides are joined together by RNA polymerase. And that makes sense, doesn't it? The idea that polymerase is an enzyme that makes a polymer, and it makes a polymer of RNA. In other words, all of those nucleotides are joined together. Then our higher end students were the ones to click on that pre-mRNA is formed first, but then splicing takes place to remove the introns, that's the non-coding sections, and that then leaves mature mRNA. This detail was missed out by a lot of students, and indeed some students said that plant DNA didn't contain introns. So let's remember that it does, and just because it's asked about a plant, it's no different from asking about an animal. Hope the video's been of use. Thanks for watching. Don't forget to leave me a comment.